All right. Hey, ready to go. New material coming your way. We have a little to finish up. I got around to doing the review recording, about 35 minutes of me blabbing. So listen to that multiple times. I already see people are starting to listen to it. And so if you didn't get to see it yet, that's fine because I didn't send it out. So, last night. Sir.
So for the new exam, it's chapter 6, 8, and 9 going to be part of it.
No, this exam as was left of the cell that was on there. That's chapter effectively the three I do in the PowerPoints, but it's like 3 and 4 in your textbooks. That's the cell stuff, osmosis, facilitated diffusion, all that neat stuff about the cell, resting membrane potential, et cetera, solid cell cycle. So those are the big topics out of that particular chapter. The bone stuff, which is whatever, there's 7 or 6 in the PowerPoint sequence that I prefer to use, and then a little bit of joints. That's all detailed. The study guides there. I sent it out again because apparently the first time I sent it, it may not have gotten to everybody or to all the classes, so I did a resend of that over the weekend. So you should have seen the study guide should be there as well as now the review recording. So you should be covered. And we only have one more thing that's on it that I didn't cover. That's what we're going to do. And that's gout. The part on Lyme disease I'll comment on, but we're not going to be doing it. And then whatever I do today, starting with muscles, that's all the next exam, and so we're getting into the heavy duty equipment. This is where it really starts to get tough. And you're going to see today is all about terminology, and there is a bunch, and it only gets more confusing and more complex as we go on. On with the show. Gout. Gout is technically an arthritic condition, but its onset is, we're okay here. It's effectively a metabolic condition that presents mostly as a kind of arthritis. It can still be fatal. Okay, but nowadays we have a lot of good medications, and it's something that's relatively easy to pick up, and it's not something that, and certainly in what I would call Western medicine or established medicine, easy to find. It has to do with an overabundance of a chemical called uric acid. Uric acid we all have. It's not that you can magically not have it. Let's see if I get that cursor to work a little bit better than there. So that uric acid is something that causes the DNA, as we gobble up cells with DNA and RNA, all of those materials, as we recycle them, we create waste products. Uric acid is one of those waste products. You don't have to know it, but it comes from adenine and guanine. And so the uric acid, we always have a certain amount in our bloodstream that's in the process of being delivered to the kidneys to get rid of, in the urine. Basically, that's the way it's disposed of. What happens when we have too much of that uric acid for any one of a number of reasons? Years ago, they thought it was diet, and is diet a contributor? Yeah. Most of the time, it has to do with a genetic defect that involves an enzyme that doesn't break it down as efficiently as it should. So effectively, we have too much. So diet and lifestyle is a contributing factor, but really, to a certain extent, it's maybe 10% of the picture, 90% of the picture involves this enzyme. So when there's too much uric acid, it forms crystals. Unlike salt crystals, the crystals are like daggers, and they have a predilection, a tendency to form in joints, particularly joints in the foot, and the elbow would be the next common area. And these daggers are too big to be gobbled up by white blood cells. White blood cells come to deal with the irritation. It's like somebody's poking with a pin or a needle, and they come to the site of irritation, they spill out their contents, and it gets super inflamed. I mean, it's like, if you look at it, it looks like someone's got a real hot foot. You would think it was a giant infection inside, but the reality, it's a very acute inflammatory process, red hot and swollen. Men, roughly between age 35 and 55, certainly the majority. After age 55, it's about equal, men and women. I've treated hundreds of cases. Fortunately, with the medications today, there's medications that you can use to deal with that acute phase. And there's daily medication, just like blood pressure medication, that have relatively low problems, really low side effects. And so it's relatively easily manageable. But what happens if there's too many of those uric acid crystals, they begin to damage organs like the heart, blood vessels, the aorta, and people can literally die from gout, which is and back in the old days. Nothing new on the scene. We first saw Egyptian hieroglyphs pointing to that condition, just to give you an idea. Mankind has been suffering with that for a very long time. That's the treatise on gout. This other one we deal with in micro. For the most part, unfortunately, roughly 2 out of 3 of you are going to be stuck with me for micro. I know you can't, just your bad luck. Best advice is wait 2 years. Maybe I'll retire. The big decision will be June of next year, not next year, but June of 27. Sorry, if you can hold on, you can skip the micro. Or take it someplace else during the summer, maybe it'll let you. So Lyme disease we cover there. Lyme disease is a bacterial infection that is caused because of a tick bite. The bacteria are inside the tick. It is the number one, that's called a vector-borne disease. The vector is another living thing that brings it to you. It could be an animal or it could be an insect. Insects are technically animals, by the way. But most of these are mosquito bites, tick bites, lice, etc. That kind of stuff. So that's what's meant by a vector. And this particular vector is a tick, and why it's the number one vector-borne disease in this country has to do with an abundance of something that participates in that tick's life cycle, which is a deer. So the very famous white-tailed deer, of which we have an extraordinary abundance around here. And we have an extraordinary abundance of ticks, both because of that and because of the nature of our terrain and our climate. So all that being aside, that's why we get a bunch. So really, it used to be a skin disease because it's highlighted by a bullseye-shaped rash that most people get somewhere upper thigh, upper shoulder, that kind of area. Typically, we see that rash. And that's what I talk about when we do micro. But really, it is an immunological disease. That's now it's carried out. So just like rheumatoid arthritis, which I spent some time on, your body's reaction to this tick bite produces a lot of immunological chemicals and activates a lot of immunological cells that damage other parts of your body, particularly the nervous and muscular system and the joints. And we see that if it's not treated properly initially or if it's not treated sufficiently initially. And a lot of people, it surfaces before they know they actually had that bite. So a lot of times the bite will disappear and all of a sudden they're coming to see the doctor with fatigue and non-specific symptoms and aches and pains, and I can't move. And and when we get into micro, you learn about the different stages: first stage, second stage, and third stage. Treatment, fortunately, is antibiotics. The sooner you get it, the quicker the treatment. The longer it takes to be diagnosed, then you can go into problems which can even be permanent. So that, and I'm not going to ask a single question on this test. I'm saving that for micro. But I'm giving it to you because at least now we're starting to be included in its own type of arthritis. So that's why it sort of has a subcategory. That's all for that whole, but that's the end of anything that potentially could be on the test. And in this case, it's specifically the gout information. So if you'd like to forget about Lyme disease until next semester, you'll have my blessing. Wait, everybody, no, not a smile. God, you're not a fun group. It's me. I'm tired anyway today. Who knows? So this is where we really start the hard stuff. I'm saying it very sincerely to you. You're going to see right now, we're going to start to talk about complexity. I know you talked about muscles in lab, okay? And you talked about, you named different muscles, and maybe you talked about the shapes of muscles, parallel and fusiform and convergent and pennate, and, right? Terms like that, that's anatomy. Origin and insertions, that's anatomy. How the muscle works is what we talk about here. This is very much dedicated to physiology. So the anatomy portion is going to be much more on a microscopic level and indeed a molecular level. And that's why it's going to be very difficult. And so I sent everybody this earlier. This is rather a lengthy, big megabyte type of PowerPoint. It's got 3 brilliant animations at the end that we are going to spend a lot of time on. Most likely, we'll probably see this. The earliest would be Friday. I think it might end up more on next week, but we'll see. So this is the starting. Muscles, half the mass of the body. They're made. All muscles are giant amounts of protein, and so our bones may have. A lot of people say, well, bones are very heavy. It's not. It's about the muscles. So about half of our weight comes from our muscles. Okay, and really, well, part of it is the physiology is understanding the way we change energy from one form to another. So these energy transformations, we're going to take chemical energy derived from the ATP that we have generated, and that energy is going to effectively be used to make a change. We call it excitation. You'll get to this. Excitation, contraction, coupling. We're going to use this chemical energy to generate effectively almost an electrical current that transfers from a nerve to a muscle and causes that muscle to shorten or contract. Muscles don't bend. Basically, they shorten. And that shortening, depending on the shape of the muscle, will cause different. It's like your biceps brachii. You might remember that's a fusiform muscle. It's kind of fat in the middle. That's why the muscle bulges. By the fact that there are all these what we call fibers, and a fiber is a cell, shorten, effectively it causes distinctive shape change. So we're really looking at that, and we're going to explore how this occurs 3 ways. We're going to explore how the nerve sends the signal to the muscle. We're going to show how the muscle changes that signal from effectively chemical / electrical energy into mechanical energy, and then specifically look at what causes the muscle to shorten. So there's 3 enormous parts of this, all of which are in this very first PowerPoint section 9A. Takes a while to get. I typically, for the exam, I make it a 1-chapter exam. That's my plan for this class. So you can imagine how much content there is if it's only 1 chapter. So I'm being very sincere with everybody, and I'm delighted to see everybody here who's here, because you really need to be here and listen to this, go over it and hear it, and please use these recordings. Listen to them. I understand that you now either on Tuesday or Wednesday have a unit of free time, not currently being occupied by lab. So one of those times, every so often, listen to me. I know if you could turn down the volume, you're fine. Not a smile in the bunch. I love it. Half the body's mass transforms energy, different types of muscle we're going to look at. So we're going to look principally at the types of muscle a little bit. We're going to look at characteristics a little bit. Functions are pretty straightforward. The big deal is terminology. It's very confusing. For instance, I just alluded to one. We're going to talk about fibers in a muscle. Those are cells. A muscle fiber and a muscle cell are the same thing. The fibers, as you will see, contain elements in them, flexible tubes of protein. They're called fibrils. Not done yet. Those individual flexible tubes of protein contain within them filaments of different kinds of proteins. So and that's where the confusion is for exam taking. Why I urge you, whether it's this coming exam or that exam on muscles, read questions carefully. Because if the question says fiber or versus filament or versus fibril, it all may have a different meaning. That's why it's so important. No one's going to ask you about it in your career. But the idea of dealing with complex material, being able to have a reasonable degree of comprehension of it and be able to answer questions about it is what applies generically to your careers. And it's good to know something about how muscles work. I'm sure you wonder why we get into such detail, but there's a reason for it, and obviously the important reason is part of the curriculum. We have certain things that we're obligated to teach you to get certain degrees in certain specialties that you're entering. That's the way it is. Can't help it. Go anywhere, it's going to be the same type of thing. So myo and meso you're going to see those terms. So myology is the study of muscles. Meso, you're going to see some of the outer, and maybe you covered it in lab, epimysium, perimysium, and endomysium. Is that ringing a bell from lab? Maybe? Okay, we'll look over it again. Sarco is when we get down to that molecular level. We're going to look at, and for instance, the sarcoplasm is actually the cytoplasm of the muscle. The sarcomere is the individual repeat unit. Remember we did monomers and polymer? Well, the sarcomere, they put an E at the end of it, is an individual unit, and that individual muscle fiber or cell could literally have thousands of them all lined up in a row. And they use a very good analogy in this sequence, in this author, like boxcars in a long train. And each of those boxcars, we're going to study the contents of those boxcars and see how it literally makes those boxcars get shorter. You line up all the boxcars getting shorter, that's how a muscle shortens, and depending on its shape, it may provide flexion or extension, abduction, adduction, etc. Okay? So that's where sarco is important. Sarcomere. There's a new name for the endoplasmic reticulum. It's called the sarcoplasmic reticulum, but it happens to be smooth endoplasmic reticulum. It's a giant series of wells that hold charged calcium. This is where you're first going to start to see in nerve and muscle this extraordinarily important role that calcium plays, and you're only going to repeat it again as you get into AP2. Which thankfully, you won't have me teach you, because it would be annoying. Yet I am here to answer your questions, at least for another year and a half. My wife would prefer that it would be for a longer period of time so I wouldn't be bothering her in the house. I have you captive to use these terrible anecdotes and funny, not so funny jokes. Her, she can tell me to, well, we don't want to say that. So, skeletal muscle, voluntary, you control it. It also, and it also is striped. The stripes have to do with a lot of these, what you're going to see very briefly today at the end, called filaments, where they overlap. That overlapping effectively implies strength. So these are voluntary and they are powerful in their nature. So the command for me to grab this, do this, open this, those are all voluntary actions. Cardiac muscle, all about the heart, I spend very little time on, because you do a giant unit in AP2 about the heart. It is involuntary. You can't change the way that muscle works, how rapidly or how slowly, unless medication is used. Same for smooth muscle. So it's either, and I'm fond of saying it this way, if you want to alter the way that functions, if we don't have any obvious control, we can use medications, and some people even successfully using meditation. You can have being able to relax and control or lower your heart rate. Part of it. I don't know enough about that to teach it, sir. So cardiac is also has striations. So striations imply strength. Smooth muscle has striations, they're not visible. So the distinction, and that's where anatomy is funny. Skeletal muscle is called voluntary and striated. Cardiac, involuntary, can't control it, and striated. They are both powerful. The beauty of smooth muscle, though you can't see the striations, it has tremendous amount of endurance. It works all the time. It works constantly. The beauty of the cardiac muscle, has this unique blend of power and endurance. And as you'll hear me say, if you haven't heard me say it before, it's the muscle in your body that beats more rapidly than once a second, every second you are alive. That's pretty impressive. To give you an idea. So skeletal and smooth cells are elongated, referred to as muscle fibers. So the fiber we're going to study is the skeletal, a little bit at the end on the smooth, not too much on cardiac. So skeletal muscles have particular packaging, okay? The fibers can be as long as a foot. If you don't remember it from lab, I'll help you out with it. The longest individual muscle goes from your hip down to below your knee. It's called the sartorius. It's a parallel or a strap-like muscle. So effectively, those are the long. And so think of something that could be a foot long and be more, have a diameter smaller than a hair. It gives you an idea of how tiny these structures are on the interior, how there could be thousands of what you will come to know are sarcomeres. Okay, voluntarily, conscious control, skeletal muscle contract rapidly, some very rapidly. We will even go into at the end about classifying certain muscles, which are more postural muscles or action muscles, different types of way that they fire. And then these are the keywords that are associated with it. Cardiac, only in the heart. We're not going to talk about it too much. Yeah, blah, 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 blah. And smooth muscle, hollow organs. Think of tubes. So we have a unique tubular structure, blood vessels, genitourinary tubes that are there, whether it's ovarian, if it's a uterine tube, or a ductus deferens in the male, whether it's a tube in the kidney, and there's a bunch more tubes than anyplace else, a bronchial tube. All of those are tubes. They all are technically hollow organs because they have an opening in the middle for stuff to go through, whatever that stuff is. Okay? And so visceral meaning has to do with internal organs. Non-striated because you can't see them, and involuntary. And I mean, whatever you get out of this is fine. That's for lab. If you use those in labs, they're all very distinctive to someone. I've taught a lot of these labs. They're very, very distinctive slides, as far as I'm concerned. But you don't have to know about them. What it does, this is the unique feature. This is when you ask me the question about what's on the test about the cell, okay? That is that whole resting membrane potential. Now you're going to see why it's important. Because excitability, all cells have a resting membrane potential across the membrane. Excitable cells, it can change very rapidly. It can flip-flop, and this is where you're going to start to see it. And that's the way the signal is sent eventually to the interior of the muscle by going along the exterior. And that's the excitability. It responds to this change, and it can receive the signal and respond to it. That's how muscles effectively contract. We're going to explore how the signal gets there, how it alters the membrane, how it gets deep inside. And this assortment of very interesting structures that students find very complicated to understand. Fair warning, please. So maybe it's a blessing in disguise that now you're done with lab, rather than have to worry about lab and this at the same time. And yes, they can shorten. We talked about that. You're going to see we explore that. And the fact that they can stretch and they can recoil. Well, I'm actually going to show you today one of the proteins that makes that happen. It's a protein that's sort of structurally arranged like a spring. So it can stretch and it can contract back. And you see, and understanding the structure can maybe explain a little bit how you can basically strain a muscle to the point where now you have to let it give it a time so it can repair on the interior, and the inflammation can subside. It's the type of thing you see in sport all the time. It's almost like you're doing we were doing leaves Saturday. I mean, I got a whole new series of ouches Sunday morning from starting to work on the leaves. We have 2 giant oak trees. I should have said for extra credit we can have people come over and rake, but I wouldn't offer any extra credit. So they produce movement, all that stuff, posture, buy it, stabilize joints, okay, if it's the shoulder mostly, generate heat when you shiver, etc. And this is the anatomy part. Classic anatomy, you talk about nerve supply and blood supply and where it attaches to and all that nonsense. That's there. We're not going to do it. The amount of oxygen is pivotal, okay? And blood supply is pivotal because it needs a ton of oxygen, constantly replenishing it, constantly replenishing what it does when it has to contract a lot with energy resources, and because of that, waste products. And that's the big problem when we get to the exercise. This is the part of This is the chapter we start talking about exercise physiology and what happens why your muscles fatigue, why you have to catch your breath, why they go into spasm. And a lot of it has to do with waste products. So there's a lot of understanding that goes on because those are things you're going to encounter in your own life. And depending on who you're interacting with, if you're in a clinical field, some of the problems they may have. So this is where we actually get into the structure and the nomenclature. So maybe this is stuff that hopefully we've got at least an introduction in the lab. Things occurring in threes is very recurrent. You talked about when we look at blood vessel, you're going to see there's 3 layers that make up a blood vessel. All those wonderful hollow organs typically are in threes, except for digestive, which is a 4th. Okay, you're going to when we look at the heart, we're going to talk about epicardium and pericardium and endocardium. Epi means outside. The MYS indicates it's muscle. Peri means around, and you're going to see those are bundles on the inside. And endo are even finer bundles, and they're all made of connective tissue. The epimysium is very dense and irregular. If you buy a side of meat, sometimes you have to trim off something that's very thick and gray. We call it silver skin. If you cook it, it gets all gnarled. It's indigestible. It doesn't really have any official nutritional aspects to it when it's like that. And that is what surrounds the muscle. All of your muscles have this, particularly in the extremities. So all those wonderful names, vastus lateralis, vastus medialis, rectus femoris, gluteus maximus, they all are surrounded by this epimysium, some more so than others. And you can see they use the word, and I don't know if you covered the word fascia. What happens is, when your muscles, if they didn't have anything surrounding them, every time you took a step, it would start to bulge. So in your extremities, you have additional connective tissue that holds them better to the bone. That's called fascia. We have deep fascia and superficial fascia. I'm not going into that. That's more surgical anatomy. But maybe you know somebody who in sport, or happened to you, who had a thing called a compartment syndrome. That's when you use the muscles so much they swell against that fascia. You literally have to go in there and lance and cut little openings in the fascia for the muscles. The muscle's going to die. It's a very painful and potentially a very dangerous situation. Compartment syndromes. But just to give you some context, why this stuff is important, and you're going to know people have this problem. They just do. A lot of people, I'm going to run a marathon. And the first day they try to run 10 miles, not really good training protocol. Why is my leg killing me? And they keep doing it. They're working on a compartment syndrome. Moving on. So the epimysium, dense, irregular, like periosteum, like the capsule of a joint. Perimysium is much more fatty containing, and the endomysium even more fatty in nature. So this is what it looks like. So you're sitting there. Here it is, attached to the bone by way of a tendon. It can go either attach directly into the bone or by a tendon or by other structures. We looked at that before. This is the epimysium. That's that outer stuff that, if I think of it in the context of what you do when you go to the grocery store and you're buying meat, for instance. So if you're buying, and you can tell the cuts if you're buying a sirloin, if you're buying a filet mignon, if you're buying a strip steak, T-bone, porterhouse, all of them are based on, so they take the long part of the meat and they slice it, perpendicular to the long axis. So what you begin to see there is, when you look at this, all of these little bundles inside that are surrounded by perimysium, you can see it here, those all contain a certain amount of fat and connective tissue, and these are all fascicles. Nerve has the same appearance. When we get to nerve, the big difference in nerve, the blood vessel supply is in the middle, and in muscle, it comes from the exterior. And so another name for that in foodie circles is marbling pattern. The more fat it has, the more flavor it has. It's the difference between, if you go to the grocery store and take a look at most of the meats you see are choice, and then you look at prime, and it costs a lot more fatty in it. And if you want to buy American Kobe style or Wagyu beef, I mean, it's off the charts expensive. Not budget friendly. I'm speaking to the carnivores in the house. I have vegans in the family who I love cooking for. Moving on. So here is the perimecium. That's the fascicle. Now we're going to pull it. So we pull fascicles out. These are fibers. Those are an individual muscle cell. This is where the complexity comes in. It's a unique cell. You're born with your full allotment. You can't make any more. If they're significantly damaged, they die and they're replaced by scar tissue. It's a multinuclear cell. It's surrounded by a very delicate connective tissue layer, and that is the endomecium. And you can see these little flexible tubes. They're just chock full of protein. Those are the fibrils. So here's the whole thing. Origin insertion we're not doing. Direct or indirect, doesn't matter. Direct right into the bone. Indirect typically by a tendon or other structures that are there. And that's the kind of stuff you do in lab. I'm not asking any questions about. Same illustration. So here's the fascicle. When we pull out the fascicle, okay, we have the muscle individual fiber. It's surrounded, effectively this flexible tube, by a very delicate tissue called the endomecium. Underneath that, if we peel it away, we have the actual cell membrane. Now we use, and this is the complexity of it, now we use the prefix sarco. Once you get inside into the actual cellular layer, the part that we will be studying, the sarcolemma is really that cell's cell membrane. Lemma is a term that's sometimes used for membrane or liner in Latin. That's where they get it. So the neurolemma, sometimes you'll see that with nerve, for instance. Underneath that membrane, sarcolemma, now you have an abundance of nuclei. That's why it's a unique cell. Can't be duplicated. It's multinucleated. That's unusual. It has an abundance of mitochondria because it's very energy dependent. And if you pull out one of those flexible tubes, that's a fibril. We use the term myofibril. And what you're going to see when we look into it, I'm just going to jump ahead for a second. We're going to take that individual myofibril. We're going to look at the banding pattern. This alternates dark and light. That's where we see the striations. And we're even going to look individually. This is the sarcomere. Now we're going to spend most of the time on, not so much this time, but next time, looking at these those little blue lines there are called Z, capital Z discs, because protein-wise they have a zigzag pattern. So they just named them Z discs. And they are the boundaries of all these tiny little boxcars that shorten. So the walls come close. So all of a sudden the walls are shrinking and coming together. So I'll go back to where I was. I just wanted to give you that for some context. So this is what we were looking at before. A better blow up of that as we go forward. Long cylindrical cells, multinucleated. The membrane, we now call it the sarcolemma. If you see muscle fiber plasma membrane, I'm good with that. We don't say cytoplasm, although I'm still good with that. Sarcoplasm. There's very little of it. That cell is filled with tons and tons of protein. There's very little in that cell other than the protein rods, okay, glycosomes, which are storage containers for the energy source, which is glycogen, okay, and the pigmentation that you see is from myoglobin. Myoglobin is very similar to the way we can. It's basically how the oxygen supply, and muscles require a ton of it. The reason that muscle supply, rather, comes from a chemical called myoglobin, a unique protein with iron in it that gives it a red color. Our blood has something similar but different because of the way it releases the oxygen, called hemoglobin. Okay? And hemoglobin imparts the reddish color to our bloodstream, and that's because of the iron pigment that's in it. So if you're consuming meat and you have red meat versus white meat, they're basically the same. It's just which ones have the most myoglobin. A part that you don't use a lot, okay, does not have a lot. So thinking poultry, like the breast of a turkey or a chicken, isn't used a great deal. So it is basically white meat. Where the legs are used a lot, particularly those hind legs, the drumsticks, That's why they would call them dark meat, because they have more myoglobin. That's the distinction between the two, if you're interested. And so inside, everything is different. The fibrils are really an organelle, although unique in its own way. The endoplasmic reticulum is very specific. Again, it's a sarcoplasmic reticulum is a kind of smooth endoplasmic reticulum. It really doesn't have any rough because it's evolved to be unique for this cell. And you might recall smooth endoplasmic reticulum has a lot of cul-de-sac structures. That's for holding on to the calcium. This is where we're really going to start to see how calcium plays a pivotal role in firing the muscle and how the muscle itself contracts. And then interestingly enough, the big T stands for transverse. Transverse tubules are tubules that basically are an extension of the sarcolemma or the plasma membrane that are able to bring that signal deep inside to all of those fibrils. And these animations that are accompanying this rather generously sized PowerPoint that I sent to you, basically as an attachment because it won't work on our Blackboard, really demonstrate this wonderfully. They're exceptional. So fibrils, rods, thousands of them, 80% of the muscle's volume. It accounts for the striation. The sarcomeres are the boundaries of the individual subunits. And inside, just to get you confused some more with the terminology, so we have a fiber, we have fibrils, and each of those fibrils, those flexible tubes, are filled with filaments. Microfilaments, mostly made of actin, which you should be familiar with, because those are actin filaments we looked at in the cell, kind of things that make cleavage when we look at mitosis, kind of things that make microvilli and various, they play a pivotal role in amoeboid motion. Okay, they're here too, but they, rather than forming and unforming, they're always there. Those are one type of myofilaments. The other one is a true motor protein. It's called myosin. You're going to see it's braided. When we start talking about how proteins gather strength in braiding, and it changes shape. And in the fact, when it's changing shape, basically that's what facilitates the sarcomeres basically compacting on themselves, just like a telescope that's open, closer. So they almost telescope in on themselves. And so we're going to look at those myofilaments. See? Hard. Nothing else. I'm not trying not to scare you, but you should be at least a little bit intimidated by this because it's complex. So here's that same illustration I showed you before. Now we get to the meat of it. At least this part's easy. The stripes. You saw the stripes before, dark and light. The dark bands are called A bands. The light bands are called I bands. Allow me to point out, and this is the easy way to remember, in dark there's a letter A, and in light there's a letter I. Got it? I band, light, because of the I. A band, dark, because of the A. You can't possibly get that wrong. Those questions will be there. You remember there's no I in team, but there's an I in light. Well, I'll cover it. Yeah, you're really not going to hear a whole lot about H and M zones. I'm going to show them to you. And you're going to see the Z disc. That's important. Coin-shaped proteins that sort of zigzag. Okay? And so it's easier for me to show it to you. And this is classic view of muscle, not that important for us. Our focus point with a brief description in the next 5 minutes are the sarcomere. This is really the functional unit we spend the most time, and we're going to explore it here, and then we're going to delve deep inside of it. So effectively, I talk about an A band and half an I band. I'm going to show it to you. The boundaries are the Z discs. From one Z disc to another defines the boundary of a sarcomere. You should know that. And here it is. So effectively, look here. The blue are actin. We'll get to that, but those are the thin filaments. The red are myosin, the thick filaments. Where they overlap, it is dark from here to here. Here's a sarcomere, here to here. The dark band is in the middle of it, and the light is where just the blue or the actin is, and there's like half of an actin on one side of a Z disc and half on the other. That's if you look at the way they're explaining it, well, you have half of an I band on one side and half on the other. I mean, that's more for visual purposes. I don't know if it's exactly half, but you can see the zigzag pattern that they're representing in this illustration. So the I bands are on the really are the outer borders, and effectively, to sum it up, how this moves, how it shortens, those I bands, those actin filaments all the way to here get pulled close to the middle and effectively bring the Z discs closer, compacted. You'll see this. You'll see this with illustrations. We'll describe, and you'll see it on the animations. This middle zone, because there's a limit as to how far those blue lines or actin filaments can get toward the midline, they're sort of vacant, so you don't see anything in there. The best way to visualize it is coming up in a second. It's a very orderly arrangement: actin, thin filaments; myosin, thick filaments. Okay. As you can see, the actin is the overlap, is where the A band is. The myosin, thick filaments, they are throughout the A band, except for that middle area. And the middle area has other proteins that effectively preclude it having an overlap. You're going to see this hexagonal arrangement. And so one thick is surrounded by 6 thins. You're going to see that in a second. Easier to show it. And this is a really good illustration to give you an idea of what's in the sarcomere. I think it's the last one we look at. So here, the zigzag line or Z disc. Here, you can see the actin, that filament. And effectively, again, it's going to get pulled toward the midline. That's what's going to make this contract. They actually have a nice set of slides before and after with contraction. Here, you can see the myosin, the thick filaments. There are many here. You see that sort of a spring appearing, sort of, that's called titin. That's the protein that allows for extensibility and elasticity. That's the one that can be damaged depending on how severely you strain or stretch that out of position. Notice the overlap zone here. So when we're in the I band, you just have the thin filaments. So light goes through it easily, it's light. Compared to this area where you have, in the A band, you have a thick, and this is the hexagonal arrangement they were referring to, with 6 thin filaments around it. And notice they share. So you have thick sharing commonly with other thicks that are there. Roughly the proportion of thin to thick in this, and why it is so visible and so dark, is 2 to 1. There's twice as many thin to thick. In smooth muscle, why we can't really see it, it's about 12 or 13 to 1. So again, it's a lot of these things are optical illusions. We've given an explanation because what we see microscopically. It's that stupid lacuna. Oh, the little house for the bone cell and the cartilage cell. There's no house. It's not like an empty space. There's stuff in there. But we've just, because of the way it's stained or the way we visualized it microscopically, we've made it an assumption that probably is not terribly correct. But here, you can get the idea. It's dense. Light will not get through. In the mid areas, this midline, it's got a fancy name for the protein called myomesin. Doesn't matter. Okay, we're in this H zone because it looks like a letter H, and you'll see that a little bit better when we look at the micrographs. So that's a really important idea, to look at these cross sections. So again, the light band is here. That's the actin filaments attaching to the Z discs. That's what's actually moving and pulling the Z discs. It's not a phenomenon. It's not shrinking or expanding. It's really moving across one another. So it's all about increasing the amount of overlap. Thankfully, this PowerPoint sequence has some really good graphic images of it. And then here you can see the dark area where they at rest, they are overlapping. You'll even see when they're contracted, they overlap as well. And that's everything for today. So we're done there. We're going to pick it up. Like I said, it's going to get harder because you're going to start looking at the individual way that they're arranged in these heads. You're going to learn about some other names, troponin and tropomyosin. It is Not easy. So please stay tuned and pay attention. Keep listening. Thank you.